Okay, let's talk about the highlights of the cardiac action potential videos that you should have watched. Of course, we spent quite a bit of time last semester talking about what an action potential was. We saw it in a neuron and in a skeletal muscle. But what we want to do right now is actually talk about the cardiac action potential in the heart. Okay, so since we're at the cellular and subcellular level and we're dealing with physiology, then we're trying to understand what ions are involved in this action potential and how is it different from a neural action potential. And of course, we want to understand what's going on in both the autorhythmic cell, the cell that makes up the parts of the intrinsic conducting system, versus the contractile cells, which it makes up the majority of the cells in the heart. That's actually the cardiac muscle cells. And then, of course, we want to understand how do we send the message from one cell to another. To understand all that, one of the things we need to understand is some of our basic physiological principles of what is a resting membrane potential, what's depolarization, what's repolarization, and what's an action potential. We know the resting membrane potential is that voltage that can be measured in any cell when nothing else is actually occurring. The depolarization is what's being measured inside the cell once we have positive ions rushing into the cell. Repolarization is the voltage occurs when positive ions rush out of the cell. And of course, the depolarization followed by the repolarization makes up the action potential. So that's our basic knowledge to start with. Okay, so as a reminder of our intrinsic conducting system,
cardiac autorhythmic cells in the intrinsic conduction system generate action potentials that spread in waves to all the cardiac contractile cells. This action causes a coordinated heart contraction.
So if we compare the cells within the intrinsic conducting system, that would be our autorhythmic cells, to the cells that are our contractile cells. In other words, these are our muscle cells. Look here, we can see our sarcomeres right through here. We can see actually the thick and thin filaments represented. We can see the mitochondria supplying the ATP for this action. And so this cell is going to be connected to this cell. Now remember, where cardiac muscle cells attach to each other, that's where we have an intercalated disc. And within the intercalated discs are these specialized cell proteins that are anchoring the cells called gap junctions. But gap junctions, along with desmosomes, gap junctions not only anchor, they also allow ions to pass from one cell to another. So notice here, everywhere where we have a cell connecting to another cell, we have gap junctions, because whatever happens in this cell is going to affect whatever is attached to it, because the ions, which are affecting the depolarization and repolarization, are also going to be spread from one cell to the other through the gap junctions. So remember that we said in the last subject, we said that depolarization causes contraction, repolarization causes relaxation. Well, this cell, the actual cells, the autorhythmic cells, the cells in the SA node, the nodal branches, the AV node, and so forth, they are going to be the ones that's going to actually not contract, but what they're going to do is they're going to basically initiate the ion flow, and then that's going to be sent from cell to cell to cell. So if this cell initiates it, its ions flow here, this goes through a contraction, and which then sends the signals through, and when I say signals, I mean ions, through the gap junctions to the next cell that causes it to contract, then through the next cell that causes it to contract. Then when we repolarize, then this relaxes, then this relaxes, this relaxes. So the point is we are having a coordinated effort to cause cells to contract at a specific time in a specific order. Okay, so next let's
several cardiac contractile cells. Action potentials generated by autorhythmic cells create waves of depolarization that spread to contractile cells via gap junctions, and its effect on contractile cells.
Okay, so notice when we depolarize, that's because positive ions rushed in, so now the charge right around the membrane becomes positive because so many positive ions rushed in. Okay, so our depolarization started in our autorhythmic cell, but that spreads to the next cell. So notice that the next cell, well, let me notice then. Okay, so notice that those cells depolarized, and then when they go back to negative, they have repolarized. Well, in the contractile cell, it depolarizes, which triggers contraction, and then it repolarizes, which then triggers relaxation. So again, that occurs from cell to cell to cell in a specific order. It's just that it happens so fast, it almost seems as though they're all contracting and relaxing at the same time. But in reality, this initiates it, sends it to this cell, which causes the same reaction to occur in the cell connected to it. And that's really important because we don't want just random cells contracting whenever. We want them to contract in a way as to that we can squeeze the whole chamber shut, put pressure on the blood, squeeze blood out of the chambers. So it has to be coordinated at a cellular and even subcellular level. Now if we took a section out of this autorhythmic cell and a little section out of that contractile cell and we blew it up to look at the subcellular parts, then we can actually see what's going on with the ions. So let's do that. So notice here's the corner of that autorhythmic cell. Here's my phospholipid membrane. Here's my gap junction that's allowing ions to flow from one cell into another. So over here, this cell that we're partially seeing would be the contractile cell connected to it. Now, the other thing that we have here is notice I've got my ion channels. Notice I have a sodium channel and a potassium channel, which was the same channels basically that we saw in the neural action potential. But notice I have a third channel here. I've got a calcium channel. Now, we did have calcium channels involved, but they were mainly to allow calcium to come into the axon bulb of the neuron and cause our neurotransmitters to be released. But this is a little different in looking at the calcium channels here, because the sodium channels in the autorhythmic cell are going to be sodium leak channels, meaning they're always open. So as long as there's more sodium outside than inside, it's going to leak in. Our potassium channels are going to be there to allow potassium, because we always have more potassium inside of a cell, potassium to then escape, but they are a voltage channel, so we're going to have to reach a certain voltage before they open. And then our calcium channels are also a voltage channel, so they're actually going to be our depolarizer, because this is going to be what's called a fast calcium channel, where calcium rushes in. Now there are three very important steps that are occurring in the initiation of the action potential in our autorhythmic cell. The first one is the pacemaker potential. The pacemaker potential also is what is responsible for what's called spontaneous depolarization. In other words, it's what's going to initiate everything, and it's the reason why we actually don't have to have a neurotransmitter to open up the gate. And the reason for that is because we have enough leak channels, sodium leak channels, to where there's always going to be a leaking in, at a gradual pace of leaking in of sodium into the cell through the leak channel. So pacemaker potential is going to be tied to the sodium leak channel. Okay, now the action potential, which is always depolarization followed by repolarization, the action potential starts basically when the calcium ion is opened up, but it is a voltage gated channel, so it will not open up until we reach a specific threshold voltage. Okay, and what allows us to reach that threshold voltage is actually the leaking in of sodium through the sodium leak channel. So let's watch what happens on that. Okay, now notice I have my time sequence over here in milliseconds, thousandths of a second, but I have my voltage here. So we're going to start out at a resting membrane potential, which is around negative 60 millivolts. So when nothing's actually going on here, I'm starting out at negative 60. Now, I say that, but we always have leak channels, so sodium's always going to be potentially leaking in till we restore it to that resting membrane potential. But we got to start somewhere, so let's say we're starting at this negative 60 resting membrane potential. Now, also notice that my charge on my membrane, which is why we get the term polarization, because I have an opposite charge on one side than the other, like I have opposite pole, north pole versus south pole. Okay, so Again, I'm at negative 60, so I'm negatively charged inside. This is outside the cell, so there's a lot more sodium out here, so it's going to be positively charged in relationship to the inside. So when I click on this and sodium starts coming in, watch how this gradually rises as sodium gradually comes in. What we're wanting it to do is rise to about negative 40, which is going to be our threshold voltage, which causes our calcium channels to open up.
Begin depolarizing due to potassium. Click the sodium channel to begin this slow.
Notice sodium coming in, sodium's gradually coming in, so my voltage is gradually
As sodium
rising.
ions enter the cell, the inner surface of the plasma membrane gradually becomes less negative, generating the pacemaker potential.
So this part of my graph here is called the pacemaker potential. It's due to sodium gradually coming in. So the positively charged sodium has rushed in. We've caused our membrane potential to go from resting up towards 40. Once we reach 40, that is the threshold to open up our calcium channels. So we're going to look at that next.
When the membrane potential gets to minus 40 millivolts, it has reached the threshold for initiating an action potential. Calcium channels open, and positively charged calcium ions rush into the cell. Click the calcium channel to begin calcium influx.
Okay, calcium
Note
influxes.
on the graph that the calcium influx produces the rapidly rising phase of the action potential, depolarization, which results in the reversal of the membrane potential from negative to positive inside the cell.
Okay, so let's look at that for a second. So calcium, once we reach negative 40 due to the influx of sodium, calcium channels open up, calcium rushed in, and that's why this was steeper. This was more gradual as it leaked in, but calcium rushes in. Notice though, as soon as we cross 0, now look, our charge is positive. Because what's above 0? Everything positive. What's below 0? Everything negative. So as soon as we cross that, and the term for that, which you need to know, is called reversing membrane potential. As soon as we reverse membrane potential, what happened was that was the kind of the trigger for the potassium channels to open. So when the potassium channels open, then we're going to get into our third, which is going to be our repolarization, where our voltage is going to drop again back to our resting membrane potential. So let's watch that.
To see the potassium efflux and the resulting repolarization, click the potassium channel. The potassium efflux produces repolarization, bringing the membrane potential back down to its resting level. Although not shown here, several other events take place during repolarization.
Okay, so we had our repolarization. Now, something else very important. I wish I would have shown a graphic to show this, but it's very important. Sodium-potassium pumps are activated, so sodium will be pumped back out and potassium will be pumped back in. That will keep them from reaching equilibrium on both sides so we can continue to do this process. We will also have calcium pumps that will pump calcium out. Now, but one thing that happens is that some of the sodium and the calcium, instead of being pumped out, some of it will actually start crossing across the gap junctions into the adjacent cells. And that's real important because that's what's going to initiate our action potential in our contractile cells, as we will talk about next. So here we have our three parts of our graph, our pacemaker potential, our depolarization, and our repolarization. Again, pacemaker potential due to our sodium leak channel, sodium leaking in. Once it reaches our threshold of negative 40, calcium channels open up, calcium rushes in for depolarization. Once we reverse membrane potential, in other words, we go from being negative to being positive, then that causes our potassium channels to open up and potassium effluxes, causing our returning repolarization part of the graph.